Hello, Mr. Allman here, and this is your Allman Cast on cell division. One of the fundamental characteristics of life is that all things need to be able to grow. Now, for multicellular organisms like ourselves, growth is accomplished by dividing our cells, increasing them in number, and thus becoming bigger. So, cell division is a tightly controlled, regulated process which is going to ensure that the right cells divide at the right time. Now, there are other reasons why cells can divide. We see that single-celled organisms, from bacteria up through eukaryotic organisms like amoebas, will go through a process of cell division in order to replicate. It's a form of asexual replication. We know as well that when cells are damaged, they may go through a programmed cell death, and they need to be replaced so that the function of those particular cells are not lost. And so cell division can replace those damaged and dead cells. So we'll go through first the cell cycle and then the specific process of mitosis that's going to involve the division of DNA. So first in the cell cycle, the quick review of the phases of a cell's life. As the cell is in its infancy, it goes through the G1 phase. There's a significant amount of growth that happens during this time. The replication of new organelles, an increase in size, and the cell's also going to be ramping up to be able to accomplish whatever its life function is. Now, in many cases, that cell will stop here in the G1 phase and not move on to the next phase. When it's in that senescence, it is going to be in the G0 or non-dividing state. Most of your cells spend most of their time in the G0 phase. However, if there is the right signal for a cell to be able to move on to the next stage, then it will go enter into the S phase. The S phase is when the DNA is going to be replicated in preparation for cell division. Once a cell enters into the S phase, it's likely that it's going to continue up until the point in which it's going to divide. After DNA is replicated, the G2 phase is going to be where it prepares for division. There's going to be a little bit more growth. There's going to be the formation of some new cell parts that are going to be necessary to accomplish cell division. And then finally the M phase, which is when mitosis itself occurs. And at the end of mitosis, the cell will divide, and so we have two daughter cells being produced as a result of that division. Now, much of what happens during cell division is going to be ensuring that DNA is going to be divided evenly. Now, because DNA controls most every process within the cell, every cell needs exactly the same amount of DNA. There can't be any more or any less in any given cell because that's then going to disrupt the ability of that cell to be able to survive. So to do this, I remind you of the structure of DNA during this point, a highly organized, supercoiled format of DNA called a chromosome. Chromosome is made up of chromatin, and chromatin, the functional unit of chromatin, is going to be DNA wrapped around a core of histone proteins. This condenses the DNA into a very small, highly organized form, one in which it will be easily divided during cell division, and it is unlikely that any DNA is going to become tangled or lost or broken during that process. Now, eukaryotic cells, most eukaryotic cells, with the exception of some plants, have two copies of every chromosome. Each of those chromosomes is going to contain the DNA from one of your two parents. During mitosis, they're going to form in attached identical pairs because each of those chromosomes was copied during cell division. So in this electron micrograph, you can see that the centromere is the point at which those two chromosomes are joined. These are called sister chromatids because these are exact identical copies of DNA. The base sequence in each of these sister chromatids is identical because that identical copy was produced during DNA replication in the S phase. Remember also that DNA replication has doubled the amount of DNA. So when cell division occurs, we start with a diploid cell, two copies of every chromosome. After that short period where they have doubled, the resulting cell is going to also have two copies of every chromosome. So we start with a diploid cell and we end cell division with two diploid cells. Unlike meiosis, which is reductive, there is not going to be a loss of amount of DNA because of this splitting of chromosomes. Humans have 23 pairs of chromosomes. So during the G1 phase, we have a total of 46. During the G2, that's doubled to 92 because we have 2 sister chromatids for each of those original 46 chromosomes. At the end of this process, each of those 2 daughter cells will have 46 chromosomes again. So now, to go through the stages of mitosis. The names of the stages are going to overlap mostly with what you're familiar with in meiosis. However, remember, this is non-reductive, meaning that we are not decreasing the number of chromosomes per cell in the resulting daughter cells. The things that I'll describe here are going to be specific to eukaryotic cells, so the process in bacteria does work a little bit differently because obviously there is no nucleus and there are not multiple chromosomes. During interphase, which consists of, again, the G1, S, and G2 phase, there's a lot of growth that happens during those G phases, and obviously the replication of DNA is very important to be able to note. These things are both necessary to prepare for division, because the cell needs to be sufficiently large and obviously have those 2 copies of DNA ready to go before mitosis can begin. In the images, you'll see several different ways to be able to visualize the process of mitosis in cells. You can see here in the newt cell, we have the DNA labeled as blue, and we have the spindle fibers in green. The other parts of the cell you're going to find in red. We have the whitefish and then the onion cells, so that gives us some experience with a different type of animal cell and obviously the plant cell, and we'll point out some of the differences between those. Prophase is going to be the first point at which we can see preparation and actual progress towards cell division occurring. The chromosomes are going to begin condensing. So you can see that as opposed to the previous slide, there are now distinct structures apparent. They are still scattered throughout the nucleus, however, we can see that DNA is beginning to condense into that chromosome form. The mitotic spindle begins to form, which again is that set of, which is a structure of the cytoskeleton. It's organized at the centrioles. Those centrioles during prophase are dividing and beginning to move towards opposite ends of the cell. The nuclear envelope is also going to begin to break down. So right now we have still a distinct circular structure, but those chromosomes are no longer going to be contained once prophase is over. Prometaphase is a name given to the phase that links together the end of prophase with the beginning of metaphase. Now, if we actually skip ahead to see where we're going, it makes it easier to understand what's happening during prometaphase. During metaphase, the chromosomes are going to align on the center of the cell. This is called the metaphase plate, or the equator of the cell. The spindle fibers are clearly attached to the centromeres of each of those sets of chromosomes. So if that is the goal, then we can see prometaphase is going to be the time period after which the chromosomes begin to condense, but start moving towards the middle of the cell. So they begin their migration. They are linked now to the spindle fibers, and the spindle fibers are organized at centrioles, which have mated to either side of the cell. So that movement, once they start to move, that is going to be prometaphase. It's going to be called prometaphase until the point at which they actually reach the center. So once they are lined up, now we call that metaphase. You can see here a structure called the kinetochore. The kinetochore is a bundle of proteins that are going to attach the spindle fiber to the centromere of the chromosome. So that is going to be the point of attachment between the cytoskeleton and the DNA. Here you can see again the microtubules and those chromosomes here in the electron micrograph. You should be most familiar with how to interpret what's going on during cell division by looking at an image such as the one here at the top. During anaphase, this is when the chromosomes begin to split apart from each other and move towards the poles or the opposite ends of the cell. They split apart at the centromere, so sister chromatids are split apart from each other, and the resulting chromosomes are being pulled along the spindle fiber towards either end. Remember that the movement here along the cytoskeleton, just like any movement on the cytoskeleton, is an ATP energy requiring process. So energy is being expended in order to actually cause these chromosomes to be able to move along those spindles. So you can see in the diagram here, the kinetochore is still intact so that the chromosome is still going to get pulled along that spindle, but the sister chromatids you can see them facing apart from each other because they have already separated, and that the centromere has broken apart. So here in the image, you can see also that connection between the microtubule, which is part of the spindle fiber, and the kinetochore, how the motor protein is pulling it along. And as the chromosome is pulled along, that microtubule is actually disassembled behind it. So as you look at images of this, it seems as though it kind of disappears, and that's because it actually breaks apart once the chromosome passes that point on the tubule. Telophase is going to be the point after which the chromosomes have arrived at either end of the pole. The nuclear envelope is going to begin to reform. The chromosomes begin to decondense, so they uncoil from that tightly bound structure, and cytokinesis begins. It should be easy to see this in animal cells because, as you can see in our newt cell, there is a pinching that begins on either end to show that cytokinesis has started. We see that the blue DNA is clearly on either end, and we no longer see those very clear chromosome structures, but the DNA is beginning to become a more amorphous blob again. Now, that is the end of mitosis. Mitosis is referring only to the splitting of the DNA within the nucleus. Cell division will include as well the division of the entire cell, which is finished with the process of cytokinesis. So the end of mitosis, telophase, is actually simultaneous to the process of cytokinesis when the cytoplasm splits. The organelles, it is essentially random. There are so many copies of all of those organelle structures all throughout the cell that the cell can divide in half, and there will be enough of each of those types of organelles in the new cell. Each of those new daughter cells is going to go through a G1 phase when any missing or insufficient structures will be replicated so that the cell can continue on performing its functions. In animals, cytokinesis is going to be accomplished by a contractile ring made up of elements of the cytoskeleton pulling in on the very center of those cells. That is going to create a pinching that is going to get tighter and tighter and tighter until the plasma membrane splits, and we end up with two daughter cells, each with its own nucleus and full set of chromosomes. For plants, the process is a little different because we're going to get vesicles actually bringing in the materials and depositing materials to make something called a cell plate. That cell plate is going to fuse together with the existing cell wall on either end to be able to create that cellulose-laden barrier in between those two daughter cells. And so the formation of that new cell wall is what is going to mark the end of cytokinesis in plants. As you look at the electron micrograph, we can spot some of the different stages. You can see prophase at one, like what I'm highlighting here towards the top left, because here it's clear that there are structures, but there isn't any organization to them. The point at which they start to move and to organize in some way is going to be prometaphase. So that could be demonstrated by towards the bottom left here, or towards the bottom right. There begins to be some organization to them, but they're not clearly in the middle. Metaphase is going to be like one of these here towards the right, or one on the bottom, when the chromosomes are all clearly in a line in the center of the cell. Anaphase is going to be when those chromosomes have just moved apart from each other, but there's not a very big separation between them. So you can see early anaphase here with these two cells in the center row, and then late anaphase is going to be represented by what we see just above that, where they are just about two opposite ends of the cell. Telophase is going to be when there is a clear separation between them, between those sets of chromosomes in the beginning of the nucleus forming back around them. So like what you see here at the top in the middle and on the very bottom right. Much more separation between them, and because in this particular image we can't see very clearly what's going on with the plasma membrane, we have to use that distance as a way to be able to interpret that it's actually telophase. Thank you for watching, and please let me know if you have any questions.